Updates on the status of Tridacna crocea in the Philippines with notes on molecular approach analysis. Authors: Jane Abigail Santiago, Raymond Vincent Castillo, Lota Creencia, and Maria Carmen Lagman. There are 8 existing giant clams in the Philippines. The giant clams are included in the IUCN Red List of least concerned, vulnerable to endangered species. This is due to the overfishing for food consumption, ornaments, and aquarium trades. One of the giant clams is the Tridacna crocea. This is commonly called boring giant clams. Due to its smaller size, shorter reproduction period, and faster growth development, it is recommended for community aquaculture industry. In order to achieve this, identification of the sites where it can be found in the Philippines is essential for seeding purposes and target stocking areas. This study lists the sites of existing Tridacna crocea in the Philippines, based on the previous literatures. Sample tissue collections were also being done by this study in Batanes Island, Honda Bay, Ulugan Bay, and in Puerto Princesa, Palawan. Giant clam tissue collection were done through scuba diving, followed by DNA extraction and polymerase chain reaction method for DNA barcoding. Nucleotide blast feature in NCBI GenBank was used to identify the DNA sequences. Megaten software was utilized to produce neighbor joining and maximum parsimony trees. From 1986, occurrences of Tridacna crocea has been observed within the Philippine reefs. A total of 18 studies has been done with Tridacna crocea that includes morphological and molecular characterization, distribution, and genetic structure. The first record of Tridacna crocea in Batanes Island is presented by this current study. High densities of Tridacna crocea has been repeatedly reported into Bataha Reefs, Palawan. Most of the survey and genetic studies were also conducted in Palawan regions. The collection sites checklist has shown that Tridacna crocea has a wide distribution and can be considered reasonably abundant throughout the Philippine reefs. There are numerous phylogenetic trees that has been presented, but there is still uncertainty for the relationship among giant clam species of Tridacnidae. In particular, among species that belongs to subgenus Chametrachea. The species Tridacna squamosa, Tridacna maxima, and Tridacna crocea are under subgenus Chametrachea. These 4 species have almost near size range and usually confusing when morphologically identified. Since this is the first record for Batanes Island, we constructed a phylogenetic tree for better identification of specimen, where Tridacna novae were added since this is a recently rediscovered species of giant clam. There are still uncertainty and debate in the recent records of Tridacna novae, including the Philippines samples. The results showed that giant clams created a monophyletic group. Tridacna crocea was more closely related to Tridacna squamosa and Tridacna maxima. Tridacna novae became a sister group to other 3 giant clams. These data showed that 4 species are ecologically similar with each other. These results will support the claim that Tridacna novae is a valent giant clam species and should be added to the subgenus Chametrachea. Molecular approach is a valuable tool in the taxonomic research, especially for organisms that are closely related. This will greatly help in conservation efforts of giant clams by possible identification of the resilient population for aquaculture purposes. Thank you very much. For more information, please contact Jane.Santiago at dlsu.edu.ph and Maria.Carmen.Lagman at dlsu.edu.ph.